Next, we'll perform Lab 13, Concepts in Acid-Base Titrations. A titration is very important in a lab. It helps us to standardize solutions that we may not otherwise know the molarity of, and that's generally what it's used for. Let's click on the link, Concepts in Acid-Base Titrations, to get started. The first thing we'll do is note the initial pH and initial color in order to submit those answers here. So here we have our titration setup. You can see a burette here. Okay, in order to release sample from the burette, we'll pull down on the orange stopcock. And we have the solution that we plan to determine the molarity of in the beaker. We also have a pH meter giving us the pH in the beaker. As we add solution from the burette, the pH will change. So we have acid in our beaker. We'll be adding base from the burette. We know the exact molarity of the base from the burette, and by knowing the volume, we can calculate the molarity of the solution in the beaker. That's not what we're actually doing in this lab, though. We're only just performing the titration here and answering a few simple questions. The pH is 0.91 or 0.92, and our solution is yellow. So let's put that into our lab notebook. Let's click on the Save Lab Image button and wait for that to save. And let's put pH 0.91. All right, and color initially yellow. All right, and so now we're instructed to, we have 0.3 molar sodium hydroxide in the burette and 0.3 molar HCl in the beaker. We also have an indicator that will change color when we reach the equivalence point. So let's put this, let's put 0.3 molar NaOH, 0.3 molar HCl, and our indicator is initially yellow. Let's click back here and see if we can see something that's yellow at a low pH. So if our pH is our 0.9 and we're yellow, we could have methyl violet. But let's go ahead and keep going. I'm not sure if that's what this is. So we're supposed to dispense 35 milliliters of solution from the burette, and we're going to save our graph and we're going to make some notes. Okay, so live data, save data, and we'll dispense 35 milliliters. So let's pull down. Now we can slow this down. We have 3 different stopcock positions. We can go very slow. We can go a little faster. We can go, I guess we have 4, and we have completely open. We're going to dispense 35 milliliters, but when we get a little closer to 25, we'll slow down. All right, let's slow down a little bit. Because we want to observe a color change that may around the 25 milliliter mark. Whoop, it turned blue. Now let's finish dispensing to 35. All right, so we're at 35 milliliters. We can choose stop saving. Let's look at our lab book and we'll see our graph at a volume from the burette of 25 milliliters. We had a pH of 9.95. Let's put that in. 25 milliliters, pH 9.95. Actually, it was 25.02. We'll put that in. And and the color changed to blue. So we can go back and answer some questions now. All right, so what was the pH and color at the end of the titration? At the end, we're at the end now. We've got 12.42, or I do, and a color is blue. Let's click on our indicators. So, we were initially, what color? Yellow, and then we turned to blue. So maybe we had bromcresol green. Okay, maybe that was our indicator. Probably that was our indicator. All right, and examine the graph. What happened at 25 milliliters? Let's look at our graph. At 25 milliliters, we had a sharp increase because we've reached the equivalence point where the moles of base equals moles of acid. Okay, and then what causes the change? I'll let you answer that question.